Hi all, welcome to Free Life Science class. I am Purnima. Today we are going to study about forms of DNA. Okay, this is an important topic and a simple topic. If you are studying in degree classes or in MSc Life Science classes or you are preparing for any of the competitive exams, definitely this is a small topic in the syllabus. So basically, there are three forms of DNA: aDNA, bDNA, and zDNA. How these different forms are formed? So this is a question. So before going to study the forms of DNA, first of all, I am going to introduce the chemical structure of DNA first. Okay, so this is the chemical structure of DNA. So basically, our DNA is made up of base, sugar and a phosphate group. So we can say like this: DNA is a polymer made up of units called nucleotides. And what are nucleotides? The nucleotides are made up of three different components. What are they? A sugar group, a phosphate group and a base. Bases are of four types. Okay, we can see four different types of bases in DNA. Which are they? Adenine, thymine, guanine and cytosine. If you are taking RNA, then you can see uracil instead of thymine. Okay, now we can study about the bond which are formed in between these two strands. We know that our DNA is double helical. That is, it consists of two strands. So the bond that holds between these two strands is hydrogen bond. And it is very important too. So the DNA strands are held together by hydrogen bonds between the bases on adjacent strands. Adenine always pairs with thymine and guanine always pairs with cytosine. Here, when adenine and thymine forms a bond, we can see two hydrogen bonds are formed. In the case of guanine and cytosine, three hydrogen bonds are formed. So here in this picture, you can see that the hydrogen bond that is formed between adenine and thymine. So here you can see the bond formed between adenine and thymine, two hydrogen bonds, and three hydrogen bonds are formed between guanine and cytosine. So the next thing is from DNA to proteins. So you all know about central dogma. So what is central dogma? From DNA, proteins are synthesized. So the bases on the single strand of DNA act as codes and the letters form three-letter codons which code for amino acid, the building blocks of proteins. So there are two processes that are involved in this process, that is in central dogma. The first process is transcription, which helps to convert the message in DNA to RNA. After that, the process is translation, which convert this mRNA sequence into protein, that is polypeptide sequence or amino acid sequence. Okay, so this is about central dogma. So now you get a basic idea about the chemical structure of DNA. Let's move into the different forms of DNA. The first form of DNA is aDNA. Actually, this aDNA is formed from bDNA. Under dehydrating conditions, the DNA adopts certain structural changes which results in aDNA. You all know that commonly in every cell, the bDNA is seen. That is, bDNA is the common type of DNA. When this bDNA under any dehydrating condition, its stability is affected and it adopts a conformational change or a structural change which result in this type of DNA, that is aDNA. This aDNA is much wider and flatter than bDNA, and the aDNA has a right-handed helix similar to bDNA. aDNA has narrow and deep major grooves and a wide and shallow minor grooves. So we have seen some of the features of aDNA. There are more features regarding this one, but we will discuss it in the comparison chart. So from there you can clearly understand what is the difference between aDNA, bDNA and zDNA. Okay, let's discuss about some of the features of bDNA. So you can see from the structure bDNA. This DNA is the common type or predominant DNA inside the cell. This is also known as Watson and Crick model of DNA double helix because this is discovered by Francis Crick and James Watson. Okay, so this is also a right-handed helix. We already said that. And in normal physiological conditions like normal pH and salt concentrations, in normal physiological conditions, the DNA always prefers the B form, that is bDNA. It always will be in bDNA form. Okay. So in bDNA, the bases appears to be inside the core and the sugar phosphate backbone are at peripheries, and only the edges of these base pairs are exposed to the solvent. And the width of the AT and GC are 10.85 angstrom. The major groove of DNA is wide and deep, and the minor groove is narrow and deep. Okay, so let's discuss about zDNA. zDNA is somewhat different from these two forms. The first thing is it is left-handed double helical conformation. aDNA as well as bDNA has a right-handed double helical conformation, but the zDNA has left-handed double helical conformation. The next thing is this double helix winds in a zigzag pattern, hence it got its name zDNA. And the nucleotide pairs in the DNA occurs in nucleotide dimers. Hence we can see this type of shapes are formed in zDNA. zDNA have more or less flat major groove and the minor groove is narrow and deep. The zDNA has solid core at the center. Okay, so we can discuss about comparison chart between aDNA, bDNA and zDNA. From there you can differentiate the difference between these three of DNA. The first thing is the helix turn. So here in this picture you can see aDNA, bDNA as well as zDNA. These three pictures are entirely different. So we can from this figure itself we can answer some of the differences of these three forms. The first thing is helix turn. aDNA and bDNA have right-handed double helical conformation, but the zDNA has left-handed double helical conformation. And the helical diameter is 26 angstrom in aDNA, 20 angstrom in bDNA, 18 angstrom in zDNA. And what about helical pitch or height of helical turns? It is 28.6 angstrom. Here it is 34 angstrom. Here it is 44 angstrom. The number of base pair per helix turn is, this is one turn. So here you can see 10 base pairs in bDNA and 11.6 base pairs in aDNA and 10 base pairs in bDNA, and here 12 dimers or nucleotide pairs are seen in the 12 dimers are seen in the zDNA. Okay. And the helical twist per base pair is, here it is 31 degree, here it is 36 degree, here it is 9 or 5 degree. Okay. And the distance between each base pair is, that is the distance between the adjacent base pair. That is 2.9 in aDNA, 3.4 in bDNA and 7.4 in zDNA. And the base tilt to the normal helical axis is, here it is 20 degree, here it is 6 degree and here it is 7 degree. Major groove. What about the major groove of aDNA? Major groove is narrow and deep. So you can see that the major groove is narrow and deep. What about the minor groove? Minor groove is wide and shallow. So this is a major groove. This is a major groove. Here you can see the major groove is wide and narrow. Here it is the minor groove which is wide and shallow. What about the major groove in bDNA? In bDNA, the major groove is wide and deep. Here it is wide and it is deep too. What about minor groove? Minor groove is also deep but narrow. Okay. So in case of zDNA, you can see here is no grooves. That is, it is somewhat flat, more or less flat. And the minor groove is narrow and deep. Okay. So major groove will be flat and minor groove will be narrow and deep. What about the ribose sugar conformation? The ribose sugar conformation is C3-endo in aDNA, C2-endo in bDNA, but it is entirely different in zDNA, that is C2-endo for pyrimidine and C3-endo for purine. What about the glycosidic bond conformation? So here it is antiform, here it is antiform, but in case of zDNA, it's anti for pyrimidine and syn for purine. So it is different, okay, for purines and pyrimidines. Hence it forms a zigzag conformation. Okay, so this is all about zDNA, bDNA and aDNA. So we have studied the features of aDNA, bDNA, zDNA and the difference chart also. So please study this difference chart or this comparison chart. It will be very helpful for you if you are preparing for any of the competitive exam. So thank you. If you liked my video, please give me a thumbs up and subscribe to my channel if you are new to my channel, and also click on the bell icon so that you will get notification when I upload new videos. Okay, bye bye.